sea urchins, as well as sea cucumbers, and some of the larger sea urchins as well on the side of this canyon. A sea pen as well. Very large sea cucumber there.
Return to the vessel. Was he moving?
I was waiting for the ship. Okay. Because of the slope of the canyon, we can't get super close to the bottom to be able to see everything. So once we get to the bottom end or the flat part of the canyon, we'll be able to get a little bit closer to see a bit clearer what's on the bottom. More sea urchins, sea pen, and a sea cucumber.
White points up there too.
Can we see what that is? Can we see what that is?
See there?
Right there.
Yeah.
Thank you.
Shrimp in there?
Wow. Yeah. So we believe this is a different type of sea pen, but this is the first one we've seen this dive. Yeah.
Maybe
Frame grab.
More?
let's say some frame grabs.
Yeah, with a little shrimp down at the bottom. Alright, so continue on. Thank you.
Oh, that's a big fish.
A hylosaur fish and looks like a hexactinellid sponge. And lots of these small sea urchins.
You good? What type of sponge was that?
I know. Are we waiting on the ship again? Okay. Yep. So we're just waiting on the vessel to catch up with the ROV or the because the tether reminder that the remotely operated vehicle has a tether to the vessel, so we have to kind of be very close and good communication between the pilots of the ROV as well from the bridge of the vessel. So they're working to get situated so that we can get back on the bottom.
So I would say once we reach like this flattened area,
Yeah.
we can do a video transect. And if we see something super amazing, we stop.
Like, okay.
What do you think?
It's a great idea.
Mm-hmm.
It's a great idea.
I'm like,
Yeah,
I want to
we-
collect more, but fine. When Shirley gets here. So as soon as we get back on the bottom, we'll start the transect.
Yeah, well, we crossed the
Yeah.
channel. You know how far it is from the other side of the channel? Like to the waypoint you reached?
You measure it.
Side of it,
Just
The other waypoint.
other waypoint.
Yeah.
So,
700?
So you can get like that 500 on them.
Yeah, like the other ones. Once we reach like the, once we finish to go down this.
Slope. Thank you.
updated waypoint pitch.
So we're seeing more of the small sea urchins, sea pens. We have a halosaur fish.
Remember they put a, like stuff up. Stuff up, they take it out. Push forward, push forward.
We have some sea cucumbers. Sea pens. Some of the larger sea urchins as well.
Yeah, so we can
Okay.
start the,
We'll start transect.
start the place waypoints each 100 meters. So we will start processing some pattern, right? Okay. So yeah, we will do a transect here. So we will put same as the other days,
Okay.
waypoints each 100 meters
Yeah.
up to 500.
The 5 point trip.
Yes, this we will flash the lights every 100.
We're seeing lots of sea urchins still, sea cucumbers. So by doing these transects, it allows some of the researchers on board to be able to quantify the biodiversity in this area so they can have a better understanding about what's going on and potentially be able to predict with changing environments how this could adapt and cause these ecosystems to change or shift. We have a sea star.
placing waypoints each 100 meters. So is there
What's that?
That's a super combo.
Okay.
That's a super combo.
Me too. Mm-hmm. I trust me.
None of these.
And or the sections. Yes. It's okay. Shirley will be. She will be in here to collect. You better believe it. More sea cucumbers, sea urchins. Oh. Looks like a shadow of something. Probably a sea cucumber.
Big squid behind. Oh, I just missed it.
Oh, that was a squid? Oh.
You started already? You started? No lo has logueado, ¿no?
No.
De una.
¿Ya?
Sí.
We have a shrimp,
So poner
more of the small sea urchins.
Y start, ¿no? ¿Ya? ¿Ya empiezo?
Oh, and a sea pig.
Ya no empecé. Y ya hago observations todavía. Quiero hacer observaciones.
We're starting to see more of the larger red sea urchins and more sea cucumbers. Looks like a small shrimp potentially. And two different types of sea cucumbers. That might be a dead sea pan. Looks like another sea star potentially. More sea urchins. Sea pig. More of the small sea urchins and sea cucumber, and looks like a halosaur fish in the background there. You ready? All right, that's it for me narrating, and I hand it over to my colleague Kyle.
Hello everyone, my name is Kyle Foster. I am a grad student with the University of the West Indies St. Augustine campus in Trinidad and Tobago, and I'm from Barbados. And today we're exploring Trinidad and Tobago's deep sea. So if you're just tuning in, we spent today exploring the side of a channel just west of Tobago. We've been seeing a lot of different species of urchins, a lot of sea cucumbers, and sea pigs, which I think are very cute when you compare to other deep sea species. And if you want to get a sense of the size of the organisms that you're seeing on your screen, we have two dots, which are laser projections, and they're 10 centimeters apart. So that can give you a gauge of how big these organisms are. We've also been seeing lots of sargassum seaweed within the channel. And many of us in the Caribbean are quite familiar with sargassum seaweed. Would have started seeing it quite a lot from 2011. And it's just been inundating our beaches. And it makes its way down to the deep sea as well. So just behind this sargassum patch is a glass sponge. And all the spiky organisms that you're seeing there dotted around the seafloor are urchins. And coming up on the left is a purple sea cucumber, species of sea cucumber. Oh, there it goes. got a fish as well. So currently we are at a depth of about 970 meters below the surface of the ocean. And you can imagine that at these depths, there's not much sunlight. It's extremely cold. And the pressures down there can crush you if you're not an organism that's well adapted to this type of environment. So as I said earlier, we spent most of today making our way along the face of a channel. Of course, we had sites already planned out in mind, but we also are using a multi-beam echo sounder to be able to get a better understanding of the seafloor. So what the multi-beam echo sounder does is it emits sound waves to the seafloor, and these sound waves would basically bounce back and paint a picture of what the seafloor looks like. So then we would see any sorts of interesting structures on the seafloor and decide, hey, within this site, we want to explore this area. Because with structures come interesting biodiversity, typically. So as I said, within this channel, we would have documented a lot of urchins, sea cucumbers, would have seen a few fish as well. Some sponges. Oh yes, and then we made a very interesting discovery of a woodfall today as well, which would have had a lot of squat lobsters. So now we've explored one side of the channel, we're going to make our way across the channel to the other side. And of course, we're not only making visual observations of these organisms, we're also collecting some for science. We want to be able to understand down to the genetics of these organisms. We want to understand how microplastics are impacting these organisms as well. And we're continuing to see lots of sea urchins, and some fish as well, and a glass sponge. And we're essentially doing a transect across this channel at this current moment. So we wouldn't be slowing down to inspect anything at this time, but just making our way, getting a visual of what's around. You can see a shrimp there as well. So what we're doing here, exploring Trinidad's deep sea, is very important because less than 0.01% of Trinidad and Tobago's deep waters have been explored so far. And there's much that we don't know. And this would have been due to a lack of financial and technological capacity, which is the same for many small island developing states like Barbados, where I'm from. And adding to that, a study by one of my colleagues, Jamie Lee Luchin, she would have done, recently published a study that documented 451 species that were observed for Trinidad and Tobago's deep sea. But estimates put that actual number to be at over 1,600. So there's still so much more that needs to be explored within Trinidad's waters. And that means that more than 70% of the deep sea species here in Trinidad remain to be discovered. Nice sea cucumber just floated away there. I love to see how they move. And different species of urchins here in one shot. Some interesting mounds as well here on the sediment, on the seafloor. And there goes a little shrimp. Shrimp? Yeah. And then type of fish, they're on the right hand side. So we've had a very interesting time of collecting species today, and you might wonder exactly how the ROV that we're using, which is Sebastian, collects these species. So it has a series of methods that it uses. You can use a scoop, a metal scoop. It has a hose that it can use to like suction up organisms as well, or a clamp to pick them up, a claw. And then once again, if you want to get an understanding of the scale of these organisms and what we're looking at, the two green dots that are near the middle of your screen, those are laser projections that are 10 centimeters apart. So we can get an idea of just how big these species that we're seeing are. It's a sea pig there on the right, almost transparent.
That's the end of the transect.
Okay, so now we're ending the transect.
We'll just continue just looking around as we head north.
So we'll just be continuing to look around as we make our way north. Very interesting fish here.
Tripod.
Is that a different species of tripod? Ah. The other species that we would have been seeing were more like silver and black. This one is completely dark. The other species also had kind of like fan-like projections, whereas these ones look more like whiskers. So we're going to try to get a better look at that tripod fish. Oh, looks like it's swimming. Oh, yeah. There it goes. And settled again. Yeah, they don't typically move that far. Well, the ones that we've seen so far. And they also tend to position themselves into the current. So if you look at the spots, the specks of sediment, or what looks like dust floating down, that's marine snow. And then you can basically tell the direction that the current is going if you look at those. We're just getting a close-up here. Are we going to try to get a science frame grab as well?
Sorry?
Are we going to try to get a science frame grab as well?
Yeah, she's collecting frameworks.
Okay. So we're just getting some photos. I think we got some with lasers as well. Yeah, this one is beautiful.
That's good. Gonna stay here longer?
Yeah, that's good, Sonia. Yes, thanks.
Yeah.
Oh yeah, so it's now certainly my first time seeing this type of tripod fish. Very interesting. Someone said it looks like a flying fish. I mean, agreed. So now we're just over midway as we make our way across the channel. Continuing to see a lovely sea pig there and a fish. And then a particularly large patch of seaweed here, of sargassum seaweed. Looks like the area's been disturbed. So yeah, sargassum has really inundated the shores of many Caribbean islands since 2011. As you can see, the deep sea is not out of reach for sargassum. It's typically a species of seaweed that floats at the surface, untethered, and usually provides a habitat for lots of juvenile fish. Flying fish are known to also lay their eggs on sargassum mats. So then you also find a lot of juvenile dolphin fish, or mahi-mahi as well, around these sargassum mats. We've got a sea pen towards the right of our screen. So these sea urchins that you're seeing, which are basically look like balls with spikes coming out of them, are quite abundant in this channel, I find.
About the same as it was last night. I've just been given the okay to take the massive mask off.
That's good.
Dari. Have you seen anything exciting? Not the entrance.
So this cruise is very, very important for Trinidad and Tobago. It's essentially going to provide imagery and data sets that have been collected. And it will establish the first unified and publicly accessible baseline for Trinidad and Tobago's deep waters. So it'll be able to support research, monitoring, environmental decision making for many, many years to come.
Let's
Yeah.
zoom in on this to see what it is.
Okay, we're gonna zoom in on.
It's a pariah, but I'm not sure if it's a lion.
We're gonna zoom in on this coral, see what species are associated with it, which seems to be a dead coral.
There's a crinoid here.
Okay, so we see a crinoid on the coral. Sort of. Hmm. Almost red transparent-ish. And then an amphipod hanging out on the top. It's not like a brittle star, that's kind of a small brittle star. Yeah. The bits which have like the. Okay, so yeah. Like the extensions coming out would be the alive part. Yeah. Yeah, so the red organism that we were seeing is a ctenophore towards the bottom. We're just getting some science frame grabs of this. So the red kind of goopish looking thing that you're seeing is a ctenophore. So this is a coral. And then on the base, we have our lovely sargassum seaweed. So we're just going to wait for some of the sediment to settle. And much of what we've been exploring is very like muddy clay-like sediments making up the seafloor.
Bridge, Harvey.
Yeah, go ahead, Harvey.
Could you come to a stop, please?
Yeah, sure. I just stopped now.
Thank you.
No worries. Thank you.
Very interesting. So we've got some parts that are alive and some parts that are dead. You can see an amphipod there carrying on. And once again, the red bit that we're seeing is a ctenophore.
Any specific part you want to zoom in?
Not particularly. Yeah, a better look at the amphipod here.
So do you want a piece of this? Sandstone.
We're just inspecting for more analysis. Okay, we can move on now. Yeah, thank you. All right, continuing our exploration. So essentially for this dive today, we've been focusing on an underwater channel that has not been explored before. So we began on the channel floor, and then we moved towards the base of the western edge of this channel. There goes the beautiful sea cucumber. I just love to see how it moves. Lovely. So once again, if you want to... Okay, so we've got two green dots there in the middle of your screen. So if you want to get an understanding of exactly how large these organisms are, between the two green dots essentially is 10 centimeters. So that'll be able to give you an understanding of the size of these organisms. Got lots of fish in this area. Sea cucumber as well. And a myriad of urchins were found basically along the floor of this channel. What's coming up there, the almost like transparent organism, is known as a sea pig. And then we have another beautiful tripod fish as well. Yeah, this one is gorgeous. So we would have made our way around 7 waypoints, which would have been exploring the edges of this channel. We did a 100-meter transect across the channel as well, and now we finished that and we're just exploring. Another sea cucumber and I think a sea pig as well. Oh, we would have seen one of these earlier today.
Let's stay here because it seems like a sponge.
Okay. Alright, so this is a type of sponge.
But we stay because here we come running. So if we can settle here for a while.
Okay, so we'll just stay here for our sponge expert, Shirley Pomponi, to come and give her input.
No, it's okay. The thing that reverts down is, you know.
Okay, so just stay here and talk.
No problem.
Thank you.
Fine.
Right, so it's extremely important that we, stop and see if we can understand these species that we're seeing, because, as I would have mentioned earlier, not much of Trinidad and Tobago's deep sea has been explored. So it's important for sustainable management decisions going forward, understanding what types of biodiversity exists within Trinidad's waters. So it's an almost very like hairy looking sponge. We've also been collecting a lot of the species that we've been seeing.
So
Yep.
we're gonna collect this one.
Okay.
First we will fire a Niskin once the sediment is like
Settle.
settled. So we fire a Niskin and then we collect the
Alright.
So we're gonna fire a Niskin and then collect this species as a sample. And a lot of these specimens and subsamples will be going to largely national museums and cryobanks for Trinidad and Tobago to have an understanding of what exists within its deep sea, have these species on display for locals to become more connected to what exists in their waters. And it essentially will enable long-term study, which benefits both local and global scientific communities.
Do you want to wait more for the news? Yeah. No problem. Okay. I'll keep the camera here so you can see roughly what's going on.
So currently we are firing a Niskin bottle, which is able to collect water at depth. We're collecting this in particular for one of the scientists on board, Robert Cassidy, who's conducting studies on environmental DNA. So that will be able to tell us what species has passed through the area. Of course, that's a very explained in layman's terms. It's much more complicated.
Let me know when to fire, okay?
Yeah. Oops, yeah.
Double tree, right?
Okay, we fired our Niskin.
Let's do like more close-ups, please.
Oh, and we're gonna get some close-ups of the sponge, so we can get some science frame grabs. And we're gonna have some with lasers on, so we can get an understanding of the size in situ. Yeah, that's good.
Ah, não, não. Tem uns vizinhos que fez, né? Legal. Forte. It's a nice camera, eh?
Yeah, very nice.
The best. So yeah, I think
We got some with lasers as well? Or you-
She already did.
Okay, great.
Yeah.
You guys happy?
Okay, yes.
Yeah.
Thank you.
It goes to Via Box.
Go put by your box, please.
We
You
did.
have to choose C, choose V. Which one?
Yeah, which one is this?
Good?
So 9? Okay, so we've fired our Niskin, and we're going to put the sponge into one of our bio boxes. And all of the data that is being collected will be shared via international repositories and with the government of Trinidad and Tobago, becoming, enduring resources for education, research, and conservation.
Another, just on the other one.
C or D?
Yeah, anything that. I'm sorry. Without sea urchins on the lip, someone was saying, so I just want to make sure that they will not escape when we open it.
Mm.
If not, we also have BB1C.
PB1C might be best then.
If there are no, someone put in the chat that they are seeing like sea urchins on the lip.
Mm-hmm.
I don't know in which one, so I just want to make sure that
Yeah, I'm pretty sure we put them in
nothing will
A.
escape.
Yeah. Okay, yeah, so the sediment around is very soft. Trinidad and Tobago is essentially at the mouth of the Orinoco River from South America. So that's why we have such muddy and clay-like sediments that essentially make up the seafloor. So we're using our metal scoop and placing it carefully into one of our bio boxes. We have, very skilled pilots with us here today.
One more. No? Right. I'll retract the camera a bit when I open there. Extreme. Oh. Fuck. It's floating.
Yeah, for the other one. Something escaped?
No, when it
Almost.
starts to.
Yeah, so maybe we
Sea
can
cucumber?
go through,
Maybe one sea?
yeah, because the other one have like, you see the sea urchins close to the lip,
Mhm.
so maybe we go to this one, just to be safe.
Okay. I think it's gonna be 1C, no?
Yeah, really want to see.
Maybe 1C then. 1C. Just gonna retract the camera all the way here. And, it's all the way retract here. I'm gonna put the top camera down as well. And give you the light.
It's all the way out.
I'm pretty sure. Might lift the porch here. All the way. Look at this light. I think I want the phone. It's crazy.
And it's not as easy as it looks, folks, to control these arms and meticulously place these organisms into the boxes. Okay. Success. Okay. And then once again, especially for our Trinidadian viewers, many of the specimens that we're collecting will be hosted at the museum at the University of the West Indies, St. Augustine campus, the Zoological Museum. So make sure that when they are available, you stop by and see all the species that exist within your deep sea.
Right, beautiful porch.
So we have a number of scientists from Trinidad and Tobago students from the University of the West Indies, St. Augustine campus on this research cruise, being trained on the various methods of genetic sampling, cryopreservation. We've got a number of studies being conducted on phytoplankton toes, ichthyoplankton toes. So this cruise is very important for our local scientists to be able to, build that research capacity. It's an opportunity that few are able to get, and we're just very grateful. Because the skills that are learned during this cruise will essentially help small and developing states like Trinidad and Tobago to boost this research capacity.
Close to the edge, because I think it's more interesting than the sea. So they will have to probably
So we continue to make our way to the base of the other side of this channel. And we've collected many samples today. Got a sea pig there going out to view, and a lovely red urchin. And then these eel-like fish that you're seeing, like in the top left, that's a halosaur. And this lovely red shrimp that's coming up into view now is a pinoid shrimp. Yeah, darted away. And you'll find that many of the organisms that exist within the deep sea are a brilliant color of red. And that's because as you descend into the ocean, red is essentially the first color that disappears. So them being red at these depths kind of acts like a camouflage for them. So now I've come to the end of my time with you guys, and I leave you in the very capable hands of Shirley Pomponi, our sponge expert. And yeah, thank you guys.
Good, how are you doing? So I think they want to just go right up to the edge and then just go around, not up. Okay. Is that good? Okay, good. Hello, everyone. Thanks, Kyle. My name is Shirley Pomponi, and I'm a sponge biologist and biotechnologist from Harbor Branch Oceanographic Institute, Florida Atlantic University. And I'm here to kind of explore the last hour of our dive with you here off Trinidad. And we've been at it, well, the sub was launched, the ROV was launched about 10 hours ago. It took about 2 hours to get down to the bottom. We're at 1,968, 69 meters, so pretty deep. Oh my gosh, look at this. That is a sea cucumber, and is it dying? What's going on with that? Wow, it's got sargassum, so a brown seaweed wrapped around it. Oh my gosh. I don't think we've seen that yet, have we? We've seen some, not that much. Yeah. So for those of you who live along the Caribbean and even off the southeastern coast of the United States, there have been these, large mats of sargassum, which is, normal in the ocean, but with increased runoff and nutrients in the water, we've had this explosion of these sargassum blooms. And so what we're seeing on the bottom a lot is, some of the sargassum that just drifts, settles down to the bottom. That's the first time I saw it covering a sea cucumber, but others in the room here are saying, no, they've seen it before. So anyway, we're on the last hour of our dive, and we'll be heading back up in about another hour until it'll take about an hour and a half, 2 hours to get to the surface. And then we have a lot of work ahead of us for the rest of the evening. I'm kind of on the night shift too with a few other people. And we'll be taking a look at the samples that we collected over the past, I guess, 6 hours or so. And is it,
do anything with that.
okay. Back on? You're back on. Okay, looks like we lost the stream for a couple minutes, so we're back on. And you didn't miss too much. We're just zooming along the bottom now, and we're still seeing some sea urchins. We saw quite a few of those this morning, actually throughout the dive. Okay, it's an octocoral, I think. Yeah, I don't know. What do you think? Yeah. We'll zoom in and get some nice video of that. I don't know. Is that a sea pen? Yeah, it looks more like a sea pen to me too. So the nice thing, yeah, it's very cool. The nice thing about being in mission control, so we're not actually in the submersible. We're sitting up here on the ship in a room, mission control. And so I'm sitting like in the front row with the ROV crew who are doing a fantastic job at driving the sub and collecting these animals that we've been collecting during this dive. But there are also scientists in the room too. So if I don't know what something is, and I'm okay with sponges, but a little weak on the other stuff, I can turn around and say, what do you guys think this is? So in many cases, the ROV crew has seen it many times too, and I can just lean over and say, what do you think it is? So we're just getting some video of this. We're not going to collect it. Those little white shells that you see on the bottom are actually from an organism called a pteropod, which means winged foot. It's a kind of gastropod. It actually floats in the water, and these are all dead. They're just dead, the dead shells from them, and it's really an important food source for things that are living in the mid-water. So the dive today has been in a kind of a valley that we mapped out last night, and we've been exploring along this valley. And then I guess for the last couple of hours, we're kind of working on a slope on either side of the valley. But what we're gonna try and do just for the next, I guess, 40, 50 minutes or so, is move along kind of the bottom edge of this valley, and not move up the slope at all, just move along the bottom edge and see what we can find there. It's really nice. Yeah. So the ROV is tethered to the ship. There's a long cable that goes from the ship down to the vehicle, and that's what's supplying the power for the ROV to operate, and it's also what's enabling those images to be transmitted back up to the ship and to you all over the world. What's also cool is that in addition to having video monitors all around us in the mission control, is that there are monitors all over the ship too. So there's one in my cabin, there's one in each one of the cabins or staterooms, and one in the mess hall where I was having dinner. So a little while ago when I was eating, I saw the sponge, so I was able to quickly get up and run down here to mission control and take a closer look at it. So we're all able to participate even if we're not sitting in mission control. So I was mentioning that after the dive, after the ROV comes back up on the surface, we have a lot of work to do. So we've been taking some water samples and, of course, sampling the biology. And then the first thing that'll happen, of course, is we'll carefully pull the samples out of the bins and put them into buckets or trays that are filled with cold water, because remember, these animals were living in really cold water. What's the temperature right now? Let's see it. I don't see it right now. So we want to make sure that they don't get too hot. They'll be photographed. So we're taking some really nice images of them. And then after that, they go into the room where we're going to be preserving them. So we take samples for museum here at the University of West Indies in Trinidad. We're also taking samples for what's called environmental DNA. So if not from this animal, of course, we're not going to collect this one, but from the sponges in particular, Robert Cassidy, who was on earlier, is interested in looking at what DNA from other organisms have been filtered out of the water by the sponges, because sponges are excellent filter feeders. So what's being filtered out, and that can give us an idea of what has either been traveling through the water or lives close by. So it's called eDNA, environmental DNA. And then I'm taking a sample. Dr. Megan Conkling, who was on a couple hours ago, and I are taking samples to cryopreserve. So we take a very small piece and we put it in a special solution called a cryoprotectant that protects the tissues from getting damaged from formation of ice. And we then put it in a regular freezer, like at -20 or -30 degrees here, but it's just like the freezer you have on your refrigerators. And it can stay in that, and it freezes, it becomes solid, but the tissues aren't damaged because of this special kind of antifreeze that we have, the cryoprotectant that was developed by our colleagues at a company called CryoCrate. And the advantage of this particular solution is that when we're out in the field, like out here on the ship, we don't need a really cold freezer, a -80 freezer. Of course, as it turns out, the Falcor 2 has a -80 freezer, but most ships do not. And when we go in the field, it's really hard to get to labs or locations where you have a -80 freezer or you can get dry ice. So this special solution or cryoprotectant that we use allows us to just freeze the tissues. Oh, that's beautiful. That's really pretty. Periodically, for a short period of time, up to a couple, 3 months. But then when we get back to the lab, we can put them in at colder temperatures, so -80 or even in liquid nitrogen. And the beauty of this is that they're alive. So even though they're in kind of suspended animation, they're frozen, they remain alive. So when you thaw them out, they'll be alive. This is really incredible imagery here. You can see the polyps, tentacles, really, really beautiful. Yeah, it looks like a little, is it a, I don't know, I can't tell. There's another little animal on it. Yeah. Okay, so that animal that's crawling along is an amphipod, and kind of the way I can tell them apart is that amphipods tell them apart like from an isopod. An isopod is kind of flattened top to bottom, whereas amphipods are flattened side to side, and it's just crawling around, kind of picking off things to eat off of this coral. Not a lot of solid substrates down here, so anytime there's a solid substrate, something that an animal can settle on, it takes advantage of that. You can see it's kind of moving its little appendages up from the surface of the sea pen, the coral, up to its mouth. Okay. That's good. Yep, that's good. Thank you. See a sea cucumber over to the upper right. We've seen a lot of sea cucumbers. Those are animals now that are really taking advantage of all of the organic nutrients in the sediment. So they just basically crawl along the bottom and eat the sediment and get their nutrition from the microbes that are living in the sediment. And then you'll see every now and then, trails of their fecal matter, old poop trails. Yes, I actually said that. So I have family who's watching tonight from the United States. I'm not sure, I think Florida, New Jersey, Delaware, Texas. So hi, everybody. We're trying to figure it out. I think I think this is a sea cucumber, another cucumber. I'm not sure. Have we seen anything like that today? No. Wow. There's a little crab on it. So these are fairly transparent animals. So if you kind of look closely, you can see all the mud that it's been eating and that long kind of intestine inside it. the sediment is really, really fine here. So the least little bit of disturbance, it kicks it up. I mean, it's just, it really takes a special skill. These pilots are doing an incredible job.
There's a spot lobster. They photograph.
Mm. Oh wow, look at that. Look at that crab. Oh my gosh, the crab is like picking up. Oh. What is that? What is that? Oh, is that a, what is that? That looks like a barn.
He's eating a clam.
Is it a clam? I don't know.
Oh.
Is it? Oh yeah. Dead. But there's still stuff that's been degraded. Bottom feeders. What did you say, Anna?
Come on.
Come on. Eating.
Wow.
It's very cool. Just- I don't know what the name of this is. Oh wow, took a big bite there. Yeah, we're good. So one of the important aspects of this mission is to be able to document what's living on the bottom here. So we're fortunate to have excellent capabilities for that, capturing really good video and photos as well, building a nice library and inventory of animals this is an area that's never been explored before. There's so much of our ocean that has not been explored, and this area is one.
Sorry. Was that a crab on the sea? Is that a crab on the sea?
Yeah,
Is that unusual?
I've never seen it.
And it's eating the marine snow.
Yeah.
Have you left it already?
Yeah.
It's here in the ground.
You want to go back
Can we have
and
a
you want
look?
to go back and have a look at it? Okay,
Absolutely.
we're going to have another. There's some interest in taking another look at the sea cucumber with a crab on it. It's kind of unusual. We're seeing a lot of activity there. It's kind of, I know, it's kind of hard. Once you pick up and go, I know.
That's okay.
All right. All right, here we go. That's pretty good.
Still eating.
Okay.
What they're doing.
So this will give people at home time to kind of go and explore and look this up in the books and see, and come back on the chat and tell me what you think it is. We're going to be sitting here for a few minutes, I think.
Just holding on to it.
Wow.
Feathers and all right.
Mm,
Feathers and
yeah.
Look at that.
Look at he's just taking big chunks. He's hungry. You're gonna do-
Well, we were
You want to go? Okay. Okay. All right. So we're ready to. You're welcome. We're ready to. Okay. All right. All right. This is the last chance, everybody, to take a good close look. Okay. We're good. This time we really need it. We're really going to pick up and go this time. Okay, so some more sea cucumbers. Oh, and that one decided it's going to do a dance for us up in the water column. That's very cool. You'll see as we're moving along, there are tracks in the sediment, and those are caused, some of them are from these sea cucumbers as they move along. Sometimes there are worms. There are a lot of things that are living underneath that sediment and building, digging burrows. And so these mounds that you see are actually caused by animals that are living in that sediment. Pretty much everything you see that's crawling along the bottom are called, they're detritus feeders. They're feeding off of the nutrients, the particulate organic matter that's drifted down, settled down from the water column. So each night, we have a team of scientists and technicians, what's that, who map the sea floor. Oh, wow.
Resume the move.
Wow. And they produce these maps that enable us to be able to select where we want to dive the next day. So I'm looking, and I think they showed you earlier a image of the map that we're using to guide us during this dive. C10, I think. Yeah. And the lasers are way we can determine the size of the things that we're observing. So the lasers are 10 centimeters apart. That's useful not only for us here, as we're trying to kind of determine like simple things like where can we put this, but also later on when somebody else is looking at the video. Here's another tripod fish. We've seen a few of these during this dive. So later when others are using the videos to study the habitat and the animals that live here, they're able to be able to determine how big the animals are. Some of these sea cucumbers are pretty big. Sea star, again, eating that detritus, the sediment, getting the organics out of the sediment, microbes. So there's another sea cucumber, and you can see its trail that it left behind as it moved along the sediment.
Cheerio.
Coming up on another sea urchin. You've seen a few of these today. And they move pretty fast. They have what are called tube feet. Tube feet. And this one's just, it's moving along pretty quickly. It's like, oh no, there's something big coming after me. Pretty Tina 4.
Look at them flashes.
Common name for Tina 4s is comb jelly. We have 25 scientists and 1 artist who are participating in this expedition. More than half of the scientists are from Trinidad and Tobago, and that includes quite a few students as well. Then we have scientists from a number of other institutions, the Institute of Marine Affairs in Trinidad. I mentioned before, I'm from Harbor Branch Oceanographic Institute, Florida Atlantic University, the University of Calgary, Canada, Dalhousie University in Canada, and the National Museum of Natural Sciences in Spain. And we have a number of institutions who have contributed in some way, gear, sponsorship, the Ocean Discovery League, and the Explorers Club, and Pew Charitable Trusts, all in the United States, Pure Ocean in France, and the Anthony N. Sabga Awards for Caribbean Excellence. So we have a number of goals. We're doing a high-resolution mapping of the seafloor. Never been done in this area before, so these maps that we're using to select our dive sites are incredibly important. That's what tells us, you know, where it helps us to decide where we want to be able to dive. We're getting some heat flow data. Dr. Rachel Lauer will be doing that. I don't think she did that today, but she did it a couple days ago. So that long curlicue thing that you're seeing over to the right, that's fecal matter, kind of a poop stream. Identifying and characterizing the animals that we're seeing here in the bottom and in the midwater. We're also doing plankton tows, so phytoplankton, zooplankton, and ichthyo, or fish plankton, so fish larvae. We're also looking, characterizing any other, the microplastics. So we have samples that have been, water samples that are being taken and analyzed for microplastics in the environment. And of course, any kind of, or documenting any kind of trash or debris that we see. Earlier today, there was a fairly large piece of wood, a branch, with lots of things growing on it. Oh, something in a... See, now this gives you an idea, just looking at what's sticking out of that hole, of what might be inhabiting many of these mounds that you're seeing along the bottom. You're seeing a lot of the sargassum, the algae that's settled from the surface. Sea urchins gathering around trying to see if there's anything they can eat from that. So the color of those mounds is a little bit different. It's just because the animals that are making those mounds are kind of just, bringing up some of the sediment from underneath the surface, and so you're seeing a different color. That's why it looks a different color. There's a test or an empty shell, if you will, of a sea urchin. Looks like, yeah.
Like some sort of sea slug.
Yeah, looks like, some kind of sea slug. We haven't seen that before. Have we seen that today?
No.
No, this is really cool. Yeah, let's get some nice video of that,
I
please.
mean, I think we saw some like Voluti, I think it's a family, but they're
Bigger,
bigger.
yeah.
Like, yeah, like the mantle is
It's a
loading
little,
this
yeah.
one. This one is.
Okay. All right, stand by. Okay, got the video back.
Hey, Gerald. Hey, could you come to stop please?
Oh, beautiful. Wow, really nice. This is a type of snail. I've never seen anything like this before. So disclaimer, a lot of the cruises that I've been on, we've worked on areas where there's rocky bottom, and because I work on sponges and corals, things that have to attach to a hard surface are in that area. So this is a lot of this that we've been seeing over the last few days is really, a lot of it is really new to me.
I think this might be something
Right.
very, very interesting and new.
I don't know.
And I suggest in the river we can start it.
You want to slurp it? Okay. Okay, so after we get some images, we want to slurp this one up. So we have a lot of sampling devices that we can use to collect samples from the bottom. You've seen the claw, and you've seen a scoop, and we're going to use a suction sampler to suction this into one of the buckets.
Going to the bucket number 3. Which one?
Bucket number 3? Okay.
Number 3.
All right. Eugenie, this is going to be bucket number 3. Yeah, the suction sampler. Okay?
Bon.
What's the sample number?
049.
049? Okay. So while I'm narrating, we have someone who's logging or recording the data and snapping pictures, taking more video. Right now, that's Eugenia sitting back there doing that. It's tiny, beautiful. Can you see it? So there are lots of cameras on the ROV. There's a camera that's focused on the suction sampler, which is kind of like a rotating, several little buckets or bins. Trying to see if it made it into that suction sample bucket. Can you see it? Yeah, it's there.
Got it.
You got it? Okay, cool. Nice job. We don't need to rinse. We don't need to rinse out the sample now. You want to rinse out the sample or no?
Yeah, also,
Yeah.
but it's also
At the end. Yeah. Yeah. Okay. So the buckets have lids on them, so I'm sure it'll be there when it comes up.
What direction we are going after the?
What direction you want to go now?
We go.
What direction you want to head?
Up to
We'd like to, I think we don't want to go upslope, we want to kind of go around,
right?
right?
yeah, just
Stay
stay at the same depth.
Yeah, stay at the same depth and just go around rather than going up. Yeah? Okay. So we've come to the edge of this valley and we want to just kind of move along the edge rather than head up slope. This seems to be an area that's pretty rich with there's more, it seems like there's more down on the bottom of this valley than there was going up the slope.
What?
Mm-hmm.
We are ending here.
We're going to end here?
Yeah.
Okay. Okay, so our watch lead, Bia, has just informed me that we're going to be ending the dive now, and the sub will be making its way back up to the surface. We are at 1,961.5 meters, so pretty deep. So we'll be diving again tomorrow. Not sure exactly what time we'll be getting in, but probably around, I don't know, 9:30 or 10 o'clock, same as Eastern time, same as today. And I'll probably be narrating an hour of the dive tomorrow as well. So I hope you enjoyed today's dive, and you'll join us again tomorrow so we can explore the deep waters around Trinidad and Tobago together. Have a great evening or morning or wherever it is that you're watching or listening from. Bye.